Exploration shows how circadian control of glucose creation is disturbed during cellular breakdown in the lungs movement. New exploration from the University of California, Irvine uncovers how the circadian guideline of glucose creation in the liver is lost during cellular breakdown in the lungs movement, and how the subsequent expansion in glucose creation may fuel malignancy cell development. The new examination named Glucagon directs the strength of reverb alpha to tweak hepatic glucose creation in a model of cellular breakdown in the lungs-related cachexia, distributed today in Science Advances, represents how the circadian clock is managed under states of pressure, for example, during cellular breakdown in the lungs movement and malignancy-related tissue squandering infection called cachexia. Ladies treated with fruitfulness drugs don't have expanded bosom malignant growth hazard, study shows. New advancement could prompt a remedy for normal youth malignant growth. New immune response form conveys medications to bone tumors and restrains metastasis. Our exploration shows that a basic circadian protein, reverb, controls glucose creation in the liver. During cellular breakdown in the lungs movement and explicitly under states of cachexia, this circadian guideline is lost, bringing about expanded glucose creation from the liver, said senior creator Selma Masri, Ph.D., associate educator in the Department of Biological Chemistry at UCI School of Medicine. In light of our discoveries, we distinguished that lung tumors can give informative prompts to the liver to expand glucose creation, a significant fuel hotspot for disease cells. This examination puts the circadian clock as a focal controller of glucose creation during cellular breakdown in the lungs movement and gives significant understanding toward the advancement of novel therapeutics to target reverb, to stifle malignancy cell development. We are proceeding to research the result of expanded glucose creation during cellular breakdown in the lungs movement by following the metabolic destiny of this recently produced glucose to decide whether this fuel source can drive the uplifted metabolic interest of cellular breakdown in the lungs cells, said Amandine Verland, Ph.D., and Sung Kook Chun, Ph.D., postdoctoral researchers in the Masri laboratory. The circadian clock is our inborn natural pacemaker that keeps up physiological homeostasis in all tissues of the body. Under states of pressure, the natural clock is reworked as a versatile component to keep up synchrony and balance all through the body.